What's up, everyone? My name is Arsen Aziz, and today is our first episode and our first ever time to have a video on YouTube, okay, concerning biology. And before everything, I want to tell you that every Saturday, 6 p.m. Egypt time, we are going to have a new video, okay? We aim to have a video almost every week, and we hope that you would help us in by subscribing, by sharing, by liking our videos that will be available for every week, the next week in our lives. So let's get started. And today we are going to talk about organic molecules within the living organisms. And talking about the organic molecules within the living organism, it's a branch of chemistry that we call it biochemistry. And in biochemistry, it is everything related to the chemical reactions and chemical substances within the living organism itself. Whether is that living organism humans, animals, bacteria, fungi, or any other kind of semi-living organism like the viruses itself. That's what the biochemistry goes with. And in order to understand something, all the study of biochemistry is related with the organic molecules within the living organisms. And these organic molecules itself, they are made of carbon atoms because we are organisms that has a base of life. The basics of life in its body is the carbon atom itself. So the main idea, we are carbon organisms, okay, and we are dealing with biochemical substances that is made of carbon itself, and it reacts inside our bodies. So these organic molecules, they are large macromolecules, and these large macromolecules, we call it polymers. And these polymers, they are made from tiny small molecules, and we call these tiny small molecules monomers. In order to have the monomers to make polymers, we go to a process, and that process that we goes with is called polymerization, as you can see over here. And the process of polymerization aim to collect all these small monomers together in order to get a large molecule that can interact within our body. Most of these molecules, they tend to have a similarity between each other, and the idea of having similarity between each other because they have a similar clusters of other functional groups or other groups that we call the functional groups. And as you can see over here in the table, that we have a different functional group. Okay, that's the main functional group. We have a hydroxide group, carbonide group, or carboxide group. We have a phosphate, we have an amino group, we have a methyl group. So we have a list of these functional groups, and each functional group, it can cover several type of materials and several type of compounds. Like, for instance, if we are talking about the hydroxyl, it makes a class of material that we call the alcohols. And alcohols, they tend to have ethanol, they tend to have glycerols, they tend to have sugars. Okay, and most of these things, they have a common chemical characteristics, but that's only because they share the same functional group. But saying that, it doesn't mean since we have the same functional group that we are the same organic material. No, there are totally different organic material, but they have some common feature share between them. The bioorganic molecule itself, they are separated into four large categories, and these large categories, we call them the carbohydrates, the lipids, proteins, and nucleic acids. And today, I want to talk with you about the carbohydrates. And carbohydrates are on a class of their own. They are a large macromolecules, they are polymers, and they are made of carbon, they are made of hydrogen, and they are made of oxygen. How we did know that? Chemical analysis. And we gave them the name carbohydrates to somehow mention what is inside these molecules. So the first syllables of the word carbo, it mainly considered for the carbon, and the hydrate, it mainly goes for the hydrogen and oxygen. Okay, so the carbohydrates, generous source of energy, and they have a ratio of the carbon and the hydrogen and the oxygen itself of 1 to 2 to 1 ratio. Okay, and they have a general formula for this, which is, as you see, CnH2nOn. The N refer to the number of atoms within here. So we have a ratio 1 to 2 to 1, 1 carbon, 2 hydrogen, 1 oxygen itself. Only we should know that the 1 gram of carbs, when it is burned within the calorimeter, which is a device used to measure the amount of energy, we get 4 calories out of 1 gram of carbohydrates. They also serve as an energy storage in the plant in the form of something we call starch, we'll talk about later down the road, and also it is found in the glycogen in humans. Talking about humans, glycogen mainly stored within the liver and mainly stored also in muscles. That's what causes people to get a little bit overweight. Okay, and also we're going to talk about that later down. Now, it helps also in building cells. Okay, we have a partition of the plant cell that is made from carbohydrates, which we call the cellulose. And cellulose mainly forms the plant cell wall, okay, and that cell wall mainly for protection for the cells. So imagine that you're making protection out of carbohydrates. Well, that's pretty nice. Also, you can find it within the cell membrane in the protoplasm, and within the cell membrane, it can serve in the function of cell signaling and cell-to-cell recognition. Okay, when the cell want to interact with each other and they want to send information about each other, or they want to receive a kind of chemical material or a medication, well, it depends on certain receptors within the cell membrane. Some of these receptors in the cell membrane are made of carbohydrates. Carbs itself, they are divided into two separate groups, simple sugar and complex sugar, or we call it polysaccharide. Now, the term saccharide is always connected to the word sugar. It's a little bit of a fancy word and a big scientific word and a biological word that we use it to express the term sugar. Okay, so saying that I have a simple sugar and on the other side I have a complex sugar, there got to be differences between each other. Well, these differences are easy to make, okay, and they are easily to notice. Now, the simple sugar, mainly they have a sweet taste, they have a low molecular weight, and they are water soluble. So having sweet taste in these simple sugar, what made them um, some kind, well, tasty a little bit. We like it. We can find it in chocolate. We can find it in sugars, and we can find it in fruits, some vegetables. Well, everything concerning to these simple sugar is sweet, and they are water soluble. Okay, that means they dissolve in water and easily, no problem in that, because they have a low molecular weight, and having a low molecular weight is the key of dissolving and being water soluble. On the other hand, the complex sugar, well, they have no taste. Have you ever tasted flour, or have you ever tasted starch? Well, it has no taste at all. Okay, the second thing related to these polysaccharide is they are water insoluble. They don't dissolve in water at all, and that what made them so easily to make doughs from them. you want to make pizza? Well, complex sugar is the answer. You can make the dough of that complex, or the dough of that pizza from the complex sugar, from flour, from the starch. Okay, and that is because they have a large molecular weight. So when you put them in water, well, they tend to get a lumpy a little bit, okay, and they tend to be somehow gooey, okay. That's why you make a dough from them, and you make tasty food out of them. So back to the simple sugar, we have two classes of these simple sugar. Well, the first one is the monosaccharide, and from its name, mono means one, and saccharide means sugar. Now we have one molecule of sugars over here, and most of them have the chemical formula of C6H12O6, but some of them, they may have one, may have three, may have four, may have five, may have six carbon atoms within these molecules. So they tend to have usually from 3 to 6 carbon atoms. Okay, so having these tendency, they tend to be isomers to each other, and being isomers to each other mean that they have the same number of atoms, but they don't have the same arrangement in the space. Okay, if you have any type of molecule, when they have the same number of atoms, they tend to arrange it differently from each other, and arranging them differently from each other, they tend to have a different properties. So we having glucose, which is the king of monosaccharide, okay, and we have also the fructose, which is the sugar that we found in fruit, in honey, and some type of vegetables, especially the root one, okay, and we also have the galactose, and it's a kind of sugar that we find it mainly inside the milk, whether it is a grazing mammal milk or the human breast milk. So all of these, they have the same number of atoms, but they have a different arrangement. So they give rise to different properties from each other, okay? And like this, as you can see over here, now the glucose is the main source of energy because our cells like it so much. They tend to change and transform everything that we eat and we feed on into glucose because well, we want to make energy, and that's the simplest type of energy that we use, and that's our favorite type of energy and our favorite molecule that we can get energy from. Okay, on the other hand, this is the fructose, and it is found in fruit and vegetables and honey. And as you can see over here, now there is something about it, okay? You can find that they almost have the same number of atoms. If you start to count, you'll find that they have the same number of atoms. But having the same number of atoms doesn't mean that they are the same. As I told you before, they are isomers to each other, and though they tend to have a difference in the properties from each other. Now, that's the thing with monosaccharide, okay? They tend to be one molecule, 3 to 6 carbon atoms, found mainly, no problem with it. Cells like it, cells adore it. Well, you get a pretty much nice amount of energy from. But what happened when two monosaccharides, they are connected to each other? Now, we get, okay, that's the molecules of glucose, we get a disaccharide. A disaccharide, from its word, it contains two sugar, and these two sugar, they are connected to each other by a dehydration reaction. Okay, and a dehydration reaction mainly means that we are, in a very simple term, we are removing a water molecule. Okay, and removing a water molecule out of the disaccharide give rise to a type of bond, and that type of bond, we call it a glycosidic linkage or a glycosidic bond. Glycosidic bond is mainly related to a bond that rises between two monosaccharide sugars. Okay, So if we have here, for instance, we have a monosaccharide subunit over here. We have two, and they are mostly glucose, and they are connected by removing the water molecule. As you can see, the water molecule is exiting right here, and a new bond is forming, okay, and we call this a glycosidic linkage or a glycosidic bond. We have a ton of example about it, okay. We have lactose, which is the milk sugar, and it is mainly made from glucose and galactose, and is found in the milk of mammal, cow milk, and human breast milk, and it's also made by a dehydration reaction, removing water and forming a new glycosidic bond. Okay. Also, we have sucrose, the table sugar, mainly formed from glucose and fructose, which is the vegetables, fruit, and the and the vegetable sugar and the fruit sugar, and it's found mainly in sugar cane, sugar beet, and because they contain the highest content of sucrose. Also, the third type of sugar, we have the maltose. Now, the maltose is mainly made from two molecules of glucose, and it's found mainly in molasses and malt. Okay, so now how we can separate these two disaccharides from each other? That's simply by adding water. Okay, and that is called a hydrolysis reaction. Now, in order to test for this simple sugar presence within any food sample that we have in our hand, well, the key let's say chemical reaction that we use is the Benedict test. Now, the Benedict reagent is a blue chemical substance. Okay, we're not going through what it's made of. We're gonna go through what it's made of later. But mainly, you need to know it is used in detecting reducing sugar or simple sugar, and it is blue. In the case of you have a simple sugar that is present, now this blue color will try to change from being blue, and it will go across the colors until it reaches an orange-red color. And the darker it gets, the higher the concentration of the reducing sugar. Proof of that, we have the following image about the Benedict reagent, okay, and the Benedict test. Okay, now at the beginning we have a blue solution, no reducing sugar, no simple sugar are detected. Now it's turning a little bit of greenish yellow. Now there are traces, now red or orange red precipitate till brick red precipitate. Now you have a larger amount of reducing sugars. And as we said, as you go through the test, you need to understand the coloring, okay? Because the darker you get, that means you have a higher concentration, okay? And also another note, that you need a water bath, okay? You don't need to just put things together and leave it. No, in the Benedict test, you need a water bath in order to get the result that you want. It's a kind of, let's say somehow, make the reaction a little bit faster. Now to the big thing, to the complex sugar or the polysaccharide. Now, one of the examples of the complex sugar in the polysaccharide is the oligosaccharide. Now, oligosaccharide is mainly made from 3 to 9 monosaccharide, and these 3 to 9 monosaccharide, they are connected together by a glycosidic bond. Okay, that means that we are removing water molecules between these 3 to 9 monosaccharide connected to each other. And one of the most famous examples about oligosaccharide is the galacto oligosaccharide, which is mainly found in the soybean. Okay, so yes, soybean contains sugar. That's why it sometimes leads to the increased mass and increased weight of people. The second thing is the polysaccharide itself that grows beyond 9 monosaccharide in the chain. Now, it tends to be a slightly larger chain with branches. So, if you tend to see these small, round, beady circles over here, now imagine that one of these circles is a monosaccharide, and these dashes between them is the glycosidic linkage, and now you have a branch over here and one over here and one over there. They all connected by glycosidic linkage. Okay, now the most abundant type of food that contain complex sugar is starch. Now, starch can be broken down by intestinal enzymes. Okay, we can find the starch in the rice, potato, wheat, maize, and corn. Okay, but there are some type of complex sugar that our intestinal enzyme, well, cannot break it down, and we call it the dietary fibers. Dietary fibers, the most famous example of them is the cellulose that is found within the plant cell wall that we mentioned earlier. We aren't able to digest it or whatsoever at all. Simply can help our intestine to work a little bit better. How we can know that? Well, let's see that following diagram. I recently drew it for you over here. As you can see now, the fiber goes from the stomach, it goes through the stomach to the duodenum, the small intestine, and the fiber pass through undigested. It slows down the absorption of the glucose and maintain a healthy glucose levels. And now these small bits of fibers, it goes to the large intestine. Okay, in the large intestine, a small bit of them get broken down because we have a bacteria in them, okay, that help us to extract any kind of vitamin that they are and essentially get any nutrients or any benefit from it. But most of it, okay, it passes through and the bulk end in the stool. Also, the fibers help in decreasing the constipation. Okay, you might ask yourself why I'm getting lately a lot amount of constipation. Well, you don't eat fibers, and you can find the fibers, and you can eat the fibers when you eat fresh green vegetables. Okay, eating fresh green vegetables will maintain the movement of your intestine, the bowel movement, balance it okay and feeling great, and it will enhance your health. Okay, now carbs can be broken down by enzyme. For each kind of carb, we have an enzyme that is related to it. Well, for the polysaccharide, we have the amylase enzyme, and it breaks it down into some sugar. For the lactose, we have a lactase enzyme that simply desiccated into glucose and galactose, and we have the sucrase that break down the sucrose into glucose and fructose. We have a maltase that break down the maltose into glucose into two glucose molecules. Now, all of these sugar they goes to the intestine. Imagine that there's a part of the small intestine over here. Let's write it. That is a small intestine. Small intestine. Okay, and they get absorbed from the villi within the small intestine to the bloodstream. Now, when the amount of this glucose level increase, now it trigger the pancreas. Now, that is the pancreas. Okay, so that is pancreas over here. Pancreas. Okay, and the pancreas start to secrete insulin from a place called the islets of Langerhans within it, and the insulin instantly starts to move the glucose to the cell to get the energy that they need. Okay, and also it promotes the fat and protein production, and it simulates the liver, okay, to get working on transform the excess of the glucose into glycogen. And the excess of the glycogen they move to muscles. Okay, now how the liver work? Well, that is so easy. Now the liver work, it starts with, let's say, a molecule like the galactose, but there are enzymes that helps the liver to work, and it starts to flip this hydroxide group from its place to down here. Okay, now flipping it down here change it directly to glucose. Also, something like the sucrose and starts also will be broken down by enzyme into a 2,3 carbon molecules and sugars, well, the glucose and these three carbon sugars, they move directly to the glycolysis and then to the citric acid cycle, and from the citric acid cycle we go to something that we call the oxidative phosphorylation, and then the energy is released. All of these terminology we're gonna learn it later down the road when we talk about how the cell get energy from the food molecules that we eat. Now, the last part that we are going to talk here about it today is how to test for the starch present. Well, that is very simple. We usually use an iodine test. Okay, that procedure is very simple. You get an iodine solution and you start to put it over, let's say, a piece of potato you have, or the food sample you have. Now, if the color of the food sample start to change from the orange or the orange reddish color or the orange brown color that you have of the iodine solution, and you transform into a blue black color, now it's a positive for starch. Now, if you don't have any color change, well, that means that you don't have starch in your sample. That was all for today. Thank you so much for being here. Please don't forget to subscribe, to like, and share if you reached to the end of the video. Until I see you next Saturday, that was me, Arseni Aziz, and that was Biology with Arseni. Goodbye, everyone.